All right, this chapter is on transcription. So we're going to be talking about transcription of RNA. Now, I want to open up, of course, I always like to start our chapters off with what I would call real application, a case study, or something that you can relate to apply to what we're going to be talking about. And now, while you may think transcription does not have anything to do with your everyday life, or you don't understand how it applies to you, regardless of what your end goal is, for your career, if you are a human being, this affects you, one, not just because it is an essential component and an intermediary step to producing proteins and other RNA needed, but there are, in this example, things that can impair this process and can have a deleterious effect. In this example is the death cap mushroom. And this death cap mushroom can actually result in death if it's consumed by inhibiting the process of transcription. Specifically, it is a very potent and a specific known inhibitor that blocks RNA polymerase. This component called the alpha amidin, and it blocks both transcription initiation and elongation. And if it is blocked, it can lead to a catastrophic effect such as cell death, and it occurs because the toxin can build up in the liver, and the accumulation of this toxin building up can cause irreparable damage and functioning. So just keep that in mind when you're learning about this process of transcription, that it's more than just understanding how it works for a cell, but it has application to you. So let's go into RNA, okay? Now, we've talked about in previous slides the role of DNA and RNA, and for years, scientists not knowing what was the source of hereditary material. So for a long time, it was believed that RNA, or a portion of the time, it was believed that RNA was the sole source for genetic material. However, through studies and things that have done, there were studies that have shown in 1981, Thomas Ketch and his colleagues, they discovered that RNA can serve as a biological catalyst. For a long time, they didn't know, and this led to ribozymes, which is catalytic RNA. So we're going to talk about the primary and secondary structure of RNA. So RNA is similar to that of DNA, except it is single-stranded as opposed to double-stranded. So RNA is a single-stranded molecule, and it is composed of nitrogenous bases, but instead of having thymine, it has uracil. Okay, so again, it has nitrogenous bases. It is single-stranded, and it is composed of 4 nitrogenous bases. Instead of thymine, it has uracil. So it's uracil, guanine, adenine, and cytosine. Okay, you still have complementary base pairing, but in this case, instead of A and T, it's A and U. Okay, the structure of RNA in its linear form, this is the primary structure of RNA. It is not as stable as DNA. DNA is a double helix structure, so that double helix structure actually provides a level of stability than it does for RNA being single-stranded. Another factor with RNA is that you can have the folding or secondary structure of RNA due to hydrogen bonding via complementary base pairing. So if you have in this primary structure here, instances where you have sequences that can fold and allow for complementary base pairing, this will cause secondary structures like we see here, where you have this lariat or loop that forms. Oftentimes when you have this, it is associated with palindromic sequences, so sequences that are read the same in opposite direction, therefore allowing for this complementary base pairing that you see. So the structure of DNA and RNA, very similar, but there are differences, as I pointed out before. The sugar is a clear difference. In DNA, it's deoxy, meaning that without oxygen on that second carbon, whereas in RNA, it has the oxygen present on the second and the third carbon. Okay. The bases are clearly different. You still have the formation of 5-phosphodiester bonds in the backbone of the DNA and RNA. DNA is double-stranded, whereas RNA is typically single-stranded unless you're dealing with a secondary structure. And also the stability of the molecules are different. Again, DNA is stable. This is why you can have DNA found thousands of years later, and they're able to salvage it and sequence it, whereas RNA is easily degradable, so you don't see the presence or the prevalence of RNA as you would DNA. There are different types of RNA. Okay, so we're gonna go over just briefly some of the classes of RNA, and then this chapter will go into breaking some of them down. But I strongly suggest that you use what I give you as a guide to also go back into the book and do some investigating on your own and reading on your own. So you have ribosomal RNA, which is the RNA that is in part necessary for ribosomal processes like translation. You have messenger RNA or mRNA. This is the RNA that is carrying the message to the ribosomes to make protein. You have tRNA or transfer RNA. This is the RNA that is responsible for transferring amino acids to the cytoplasm, specifically the rough ER for translation. You have small nuclear RNA or snares. This RNA is very important in taking the processing of RNA. Small nucleolar RNA, which is also RNA that is essential in the processing of RNA. You have small cytoplasmic RNA, microRNAs, small interfering RNAs or siRNAs, which are RNAs that are what we call interfering, or what we call RNA interference, which is a process where these small molecules help to trigger the degradation of mRNA, or they inhibit translation of the proteins. And you have pWe RNAs or piRNAs. And these are another small class of RNAs that are named after what we call pWe proteins that they interact with. And you often find these in mammalian testes, and they have a role that are similar to microRNAs and siRNAs in that they help suppress the expression of elements like transposable elements. So here you have a table that I would strongly suggest, again, you study to help you understand these different types of RNA and the cell type that they're present or responsible for working with. Some are some are both for eukaryotic and prokaryotic, and some are specifically for a certain type of cell. But I also want you to pay attention to the location of these cells in eukaryotic cells. So where do they function at in a eukaryotic cell? And this is important because remember, eukaryotic cells have membrane-bound organelles as opposed to prokaryotic cells. So their role are going to differ based off of the cell type. Okay, so let's go into transcription. So just like we saw in replication, there are 3 major components. We also see this with transcription. You can divide the stages of transcription into 3: initiation, elongation, and termination. And the major components needed to carry out transcription is a DNA template, the raw materials, which are going to be your ribonucleotide triphosphates, which are going to help to build the RNA molecule. You need the transcription apparatus, which is composed of the proteins necessary for catalyzing or synthesizing RNA. Let's talk about these different components. So you have the template, okay? Unlike DNA, where there were 2 template strands, the leading and the lagging strand, or the 5' to 3' versus 3' to 5', in RNA, there's only 1 strand, okay? So the transcribed strand is the template strand, okay? And it is going to serve as the template to make the daughter RNA strand. And not necessarily the RNA strand, but the strand used to make protein. Now, the transcription unit is composed of 3 regions: the promoter region, the RNA coding region or sequence, and then the terminator region. So, based off of these, let's go over the classes of RNAs and what portions or roles they play a part in. So when you're going from DNA to RNA, that process is known as transcription, okay? And this process right here is really highlighting what we call the central dogma, okay? The central dogma. So the first portion of the central dogma is where you are going from DNA being transcribed into RNA, and that process is known as transcription. Some of the classes of RNA that are a part of that are these right here, okay? Now, some of these types of classes of RNAs are only made in eukaryotic cells, and some are made in both prokaryotic cells and RNA and eukaryotic cells, such as messenger RNA, ribosomal RNA, and tRNA. Those are both synonymous to prokaryotic cells and eukaryotic cells. However, eukaryotic cells only produce the pre-mRNA, the small nuclear RNA, the microRNAs, the small interfering RNAs. Those are unique to eukaryotic cells, okay? And then CRISPR RNA is produced by prokaryotic cells. Okay, so the process of transcription this is where we are going to be talking about in this chapter 13, right here, going from DNA to RNA. Now, if you look at this diagram here, you see that looking under electron microscope, DNA is undergoing active transcription, and it looks like what they say a Christmas tree structure, because you have this primary strand here, which is your DNA molecule, and branching off of this primary strand of DNA, you have these small branches which are indicative of RNA molecules that are being transcribed. Okay, so there are multiple molecules of RNA being actively transcribed at one time. And this is specifically an experiment that was done around 1970 using oocytes. Okay, so that's what you're looking at there. This diagram is showing you how RNA molecules are synthesized. Okay, I want you to keep in mind that unlike DNA that is double-stranded, RNA is only single-stranded. So the product of transcription should be a single strand of RNA. However, it uses DNA as a template, and the strand that is used as a template is going to be the leading strand that we saw in DNA replication, the strand that goes from the 3' direction of 3' to 5'. And that is because RNA polymerase, again similar to that of DNA polymerase, can only synthesize in a 5' to 3' manner. So the template strand has to be opposite of the direction that it is synthesizing in. So the strand is going from 3' to 5'. The RNA is going to be synthesized in a 5' to 3' direction. All right, so here you see here there are multiple genes in this DNA segment, but each gene is associated with an RNA transcript that is produced. Notice here, this is the DNA 3' to 5'. The RNA is going to be synthesized in a 5' to 3' manner. A couple of things that I want to point out when we're talking about a transcription unit, we talked about previously, you have the promoter region, the RNA coding sequence, and the terminator region. The promoter region is going to be this region here that is denoted by a negative number. Okay, the promoter region is denoted by a negative number. The transcription start site is the +1 site. This is the site that is indicative of the RNA coding region. Okay, the RNA coding region. Now, this region up here, this promoter region, is the place where all of the proteins and enzymes needed to carry out the process are attracted to. I like to tell students to think of this as the landing pad, okay, where all of the enzymes and things know to bind to. If you were thinking about, let's say, a hospital, and on top of a hospital, there's usually a landing pad for the helicopter, and it's denoted by a big red plus sign, right? This is the big red plus sign for all the enzymes and proteins that are going to carry out transcription, and they will bind here, okay? We denote them by a negative number, and each number is associated with an actual base, okay? And there are certain areas that the machinery will specifically bind to, and it's denoted by a sequence, a conserved or consensus sequence, right? But anything from going to the left, or what we say upstream, okay, away from the start site. And if it's going towards the termination site or away from the start site, it is downstream. So going to the left is upstream, going to the right is downstream, okay? And notice here, the RNA transcript that is produced is not inclusive of anything in the promoter region. So the promoter region, anything up in front of the +1 site, is not transcribed into RNA. Again, this is the DNA molecule. This is the RNA that is produced from the DNA molecule. All right, so let's go into the initiation portion, okay? The initiation of transcription, okay, starts with what we call some initiator, the ribonucleoside triphosphates, the free ribonucleoside triphosphates. It also starts with the RNA polymerase, and right now I'm talking about the bacterial RNA polymerase, and it is composed of 5 subunits. These 5 subunits make up the core enzyme. Okay, so we have the RNA polymerase, which is the core, and then we have the hollow enzyme. The core RNA polymerase is made up of 2 alpha, a beta prime, and an omega. Okay, so these are the 5 subunits that make up the core RNA polymerase. The sigma factor is the 6th component, and that 6th component makes up what we call the halo RNA polymerase enzyme. The halo is necessary for transcription initiation. You cannot have initiation of transcription without the sigma factor bound to the core RNA polymerase. Okay? Here is your triphosphate. That's the 3 phosphate groups. Know that when elongation occurs, when the free nucleotide is added into the growing chain, 2 of these phosphates are cleaved. Okay, this is the ribose, the sugar. Notice the 2nd and the 3rd carbon have the hydroxyl groups there. And then here is your base. That base is going to be a uracil, adenine, guanine, or cytosine. All right, so here you have the initiation of RNA synthesis. It does not require a primer like we saw in replication. All right. The new nucleotides are added to the 3' end of the RNA molecule, and as DNA continues to unwind at the front of the transcription bubble, you're going to have the RNA strand that is being synthesized, okay? And then it rewinds as it is completed. So as this portion of the DNA has been used as a template and the bubble keeps moving, the portion will just rewind itself. So here you have the core RNA polymerase. There's the 5 components, the 2 alpha, beta prime, and omega. Then you have the sigma factor, which is added to the core to make it the hollow enzyme. It is the hollow enzyme that is necessary for the initiation of transcription. Okay. However, in eukaryotic cells, it's a little bit different in that eukaryotic cells do not have the core and the hollow enzyme or halo enzyme. They have 5 different RNA polymerases. Okay, and these 5 different RNA polymerases transcribe different RNAs in the cell. And also you'll see that some RNA polymerases are involved in specifically plants versus mammalian cells or other eukaryotic type of cells. Either way, RNA polymerase in eukaryotic cells have 5 different polymerases. The main polymerase in eukaryotic cells for the synthesis of mRNA is RNA polymerase 2. Okay, RNA polymerase 2 is synthesizing, as you can see here, pre-mRNA. So let's go back to initiation. Okay, so you have the initiation of transcription. And what you have in the initiation component is what we call consensus sequences. These consensus sequences are these specific small sequences that have a considerable similarity between different species, and they have this similar location. And this is what the machinery, specifically initiation machinery, initiation proteins and enzymes, I'd recognize on where they should bind to begin or initiate the process of transcription. For example, the -10 consensus sequence is 10 base pairs up from the start site. So it's -10, okay? The Pribno box here is, you have this specific consensus sequence, TATAAT, okay? Then you have the -35 consensus sequence, which has this sequence TTGACA, and that sequence is also identified by the machinery. Specifically, in the hollow enzyme, the sigma factor actually saddles over the -10 and the -35 sequence. Okay? This is just showing you the actual sequence. and how you and the actual consensus sequence. So there may be small nuances that are may be a little bit different, but the core of the consensus sequence are the same. So here, just to put it in perspective visually so you can see it, here's that promoter region that is up from that start site where you have the RNA coding sequence. So if you count up 10 bases back, this is the -10 consensus sequence. From plus the site here, if you count back 35, this is the -35 consensus sequence, and it is the halo enzyme, specifically the sigma factor, that kind of saddles over this region right here and and binds there to help with the initiation of transcription. Okay, so again, this is why there is no need for an initial RNA primer, because there are initiation factors that are essential in initiating the process of transcription. Okay, and it is the location of the consensus sequence that determines the start site. So here we have the initiation. I want to show you visually how this initiation is happening. So I'm going to zoom in a little bit so you can see. Okay. And what you're going to see here is you have the core RNA polymerase. That's the 2-alpha, the beta, beta prime, and omega. And then here's the sigma factor. The sigma factor comes in and binds to the core to make the halo enzyme. And it's going to bind in that promoter region at the -10, the -35 site. Okay. Once the sigma factor binds, okay, you have the opening up of the replication bubble, and you have the beginning of the start of initiation. And what happens is you'll have a few bases that start to form a growing chain of RNA, and it's usually about 10 to 12 bases that are added to a growing chain, but it starts and stops. It's called an abortive process. So transcription will start and stop a few times, but once you have a continuous growing strain of about 10 to 12, some may say 7 bases in a growing chain, sigma factor will fall off. Sigma factor is no longer needed. Once transcription starts and you have that small growing chain of RNA, about 7 to 12 bases, sigma factor falls off, and now you transition into the elongation step of transcription. Okay, so be very clear. The sigma factor, or the halo enzyme of RNA polymerase, is only needed for the initiation of transcription. And once you have that abortive process that starts and stops, but it continues with a small growing strand of RNA, the sigma factor falls off, and now you transition to only using the core RNA polymerase to carry out the transcription of the elongation process of transcription. Okay, so again, the initiation, once the sigma factor falls off, you now transition into elongation. And elongation is carried out by the action of RNA polymerase, specifically the core of RNA polymerase. Oops. What happens in this portion right here, you're not going to have the sigma factor bound to the promoter region anymore, and now it is in the RNA coding sequence region, and it is synthesizing RNA using the template strand of DNA to synthesize a growing chain of RNA polymerase. Keep in mind, transcription is much slower, especially in bacterial cells, than that of replication. It's about 40 nucleotides per second that are added, as opposed to 100 per second. So transcription is a little bit slower than replication. And during the elongation process of transcription, supercoiling is active. Okay, so you're going to need these gyrase or topoisomerases to help carry out the process of transcription, because you're going to have positive and negative supercoiling that is occurring. All right, termination. So we know how initiation starts, we know how elongation starts, because elongation is very similar to that of replication. The difference is it's single-stranded and the enzymes that are involved in it. So there's small intricacies, but the foundation of it is the same. You have a growing chain of bases that form via the formation of a phosphodiester bond. Termination is different, okay? In bacteria cells, termination can occur in two manners, either row dependent or row independent. Okay. If termination occurs in a row-dependent manner, that means it is dependent upon the protein row. Okay. It is dependent upon the protein row to carry out termination. Specifically, row is a protein that binds to what we call a RUT sequence, an RNA sequence that is identified by the protein row, and it is a cytosine-rich sequence. And it binding there helps with the termination. Something important to keep in mind about this protein row is that it has helicase activity, okay, or ability. So it can allow the DNA RNA hybrid that is formed during the active transcription to unwind, and that allows for the ending of transcription. Now, again, let me just say the DNA RNA hybrid. That is where you have the RNA transcript, the RNA sequence that is being actively transcribed, but it's bound to the DNA template strand. So this is how you know you have active transcription, because you'll have a DNA RNA hybrid. Okay. The second is RNA independent termination, and in this case here, termination is not dependent upon the protein row. It actually is utilizing a hairpin structure that is formed by inverted repeats, and then it's followed by a strand of uracils. Okay. And so this hairpin loop or hairpin structure that forms is actually believed to cause RNA polymerase, as it is synthesizing, going down the strand and synthesizing RNA, to literally destabilize and fall off. Okay. So the RNA or row independent termination forms a hairpin structure that is thought to destabilize RNA polymerase and causing it to fall off. That is how RNA synthesis is terminated. So let's look at a pictorial representation of that. Here you have the RUT site, okay, that is cytosine rich, and there's the protein row that binds there, and the binding of that is thought to destabilize or terminate transcription, okay. And also you have this hairpin loop that is also thought to contribute to it. So once the RNA polymerase encounters this protein row, it's thought to destabilize RNA polymerase and cause termination. And let's look. So this is row dependent termination, and then row independent is where you have this inverted repeats, okay, that form this hairpin-like structure, followed by there's your strand of U's, and this hairpin loop-like structure is thought to cause destabilization of the RNA polymerase, causing it to fall off. The RNA-DNA hybrid that I was speaking about is right here. This is a DNA molecule. This is the RNA molecule that's being actively synthesized. The presence of a DNA-RNA hybrid is an indication of active transcription. All right. So we're almost done with transcription. You have in a eukaryotic cell, again, there's a little bit more complexity in that the DNA has nucleosomes that are involved, okay? And so in order for active transcription to occur, you have to have chromatin modification and remodeling to allow for transcription to occur. An easier way of saying that is you have to have the disassembling and the reassembling of the nucleosomes and histone proteins for active transcription to occur. You also have different promoters. This is another difference between eukaryotic cells and prokaryotic cells. There are multiple promoters in a eukaryotic cell that we don't see in a prokaryotic cell, and this also gives the complexity or added complexity of gene expression and regulation of gene expression in eukaryotic cells. So you have a basal transcription apparatus, but then you also have activator proteins. You have a core promoter, which has various sequences in it that are essential for transcription initiation. Okay. So, and I mentioned basal transcription apparatus. What is that? That is a group of proteins that assemble near the transcription start site and is sufficient enough to initiate minimal levels of transcription. Okay, so the basal apparatus is just enough, a group of proteins coming together to initiate minimal levels of transcription. But if you have instances where you need increase in transcription or decrease, that will be regulated by the other promoter regions. So here you have the +1 site. Okay, you have the TATA box, which is the minus 25 region. You have the minus 35, which is the recognition sequence. And then you have other regulatory promoter regions even further upstream. And all of these different regions are, the whole goal of this is to regulate transcription. Okay, regulate transcription. And the regions that I'm talking about are present in the DNA. And these sequences are used and identified by the RNA machinery to synthesize RNA. All right, so let's go into it. You have transcription and nucleosome structure. You have chromatin modifications that are going to occur before transcription. So in order for transcription to occur, there has to be modifications that occur to the chromatin so that the machinery can access it. Okay, then you have the promoter region. You have the regulatory promoter region, and this is where you have a variety of different consensus sequences that are found here to regulate transcription. Regulate meaning whether it occurs or does not occur. And then you have enhancer regions, which determine the efficiency. Okay, is it increased or is it decreased? And then you also have the polymerases that are going to bind to these various regions to initiate this process of transcription. So let's go into the initiation specifically for eukaryotic cells. Keep in mind that the process is very similar to bacterial cells, but there are some differences. And so I'm going to talk about the differences. In the initiation of transcription, you have transcription factors. Okay, transcription factors, for this example, TF2D will bind to the TATA box, okay, along with the TATA binding protein, the TBB protein. Now, one way to identify or how these transcription factors are named, TF stands for transcription factor. The Roman numeral that follows the TF is associated with the RNA polymerase that it interacts with. So in this case, TF2D interacts with RNA polymerase 2. And then the letter that follows the Roman numeral identifies the order in which it was discovered. So this suggests transcription factor 2D is a transcription factor that interacts with RNA polymerase 2, and there are other polymerases, transcription factors that were discovered before this one, A, B, and C. Okay. You also have the binding of the pre-assembled hollow enzyme that contains the general transcription factors, RNA polymerase 2, and the mediator. So you have this hollow enzyme of multiple factors that come together to initiate the process of transcription in eukaryotic cells. Okay, so we're going to zoom in and look at this process. Okay, so here you have the regulatory region, you have the core promoter region. This is going to give you the basal, the minimum level of transcription. This is going to determine whether you can have increased or decreased levels or whether it occurs at all. Here's your TF2D that binds to the TATA box along with the TBB protein. Then you have the mediator, you have RNA polymerase 2, and a host of other transcription factors that come together to form this core or hollow enzyme or a group of proteins that bind to the core promoter region and now begin to carry out transcription. Okay, a way in which you can enhance transcription is by having an activator bind to the enhancer region. Okay, and the protein binding to the enhancer region will cause the DNA to kind of loop over and bring the machinery closer to the +1 or the start site, and it thereby increases the rate of transcription. So it enhances the rate of transcription. Okay, enhances the rate of transcription. And this is the pre-assembled hollow enzyme that contains all those general transcription factors, the RNA polymerase 2, the mediator, and the other TATA proteins, TATA box binding proteins. And this is showing you how the TATA binding protein actually binds to the DNA by binding or saddling into the minor groove of DNA and kind of straddling it, that therefore allowing it to bind to the DNA. Okay, so that's initiation. Elongation of transcription is very similar to what we saw before, and that is the formation of the the processes. Now, one correction that I wanted to make is the Kornberg enzyme. Okay, so I mentioned that in our replication, but it should have been mentioned here in the transcription. Is a lot of the work that was done was through Kornberg and his colleagues, and they helped with identifying the different polymerases. Okay. So I just want to give credit to him for that as well, and not just the prior, but here in our transcription. Okay, so with the elongation of transcription, you have RNA polymerase 2 that is really helpful in synthesizing RNA. The double helix will actually enter the RNA polymerase through what we call the cleft of the enzyme. So this enzyme has different clefts or regions that bind to or allow for interaction with the DNA. And it also has components in it that help with the unwinding of the DNA. The RNA-DNA duplex, or the hybrid molecule, right, will cause it or allow it to bend in such a way, and I'm going to just go to the diagram so you can see, okay? So you can see here's the cleft here, the exit out of the polymerase. You also have where some of the other factors can come in, but ultimately you're seeing how the DNA and RNA are actually binding to or fitting into this enzyme, and this enzymatic reaction of the formation of these bases being added, how it occurs. So right, it fits right into the cleft, and that's allowing for the synthesis of these bases to occur. Okay. Again, overall, despite there being those differences, the foundation of elongation is the same, and that is the growing strand of bases by the formation of a phosphodiester bond. So now termination. There are 3 different polymerases that are found in all eukaryotic cells, and they have different mechanisms by which they can stimulate termination. So you have RNA polymerase 1, which includes that termination factor. You have RNA polymerase 2, which is going to combine or meet with RAT1, RAT1, and that induces termination. That is similar to the rho-dependent termination that we see in bacteria. So RAT1 would be synonymous with the Rho protein. And then RNA polymerase 3 is going to be responsible for synthesizing the strands of U's that we see. So there are 3 different polymerases that are thought to be involved in termination in eukaryotic cells. Okay. And we also have to consider RNA polymerase 2 and its ability to act as an exonuclease enzyme. And what it's able to do with its exonuclease activity is to attach to the 5' end of the RNA, move down the RNA, and reach the polymerase enzyme to help with the termination process. All right, so you can see here this process where you have the the RNA that is being synthesized as a function of the DNA. You have the cleavage that takes place. Here's the RAT1 protein that's binding, okay? And this portion will be degraded, and that is also thought to be important or essential in the stimulation of termination. All right, this is the end of this chapter. Just one thing to point out, transcription in archaea is very similar to that of eukaryotes more than eubacteria. So it's what that suggested that there's a closer relationship between archaea and eukaryotic cells. RNA polymerase is similar to eukaryotes rather than eubacteria. And in the promoter region of archaea, there's a similar TATA box that is found as we see in eukaryotic cells, as well as a TATA binding protein. So that is transcription for chapter 13.